Thank you very much. Um, so, so this is work that I'm going to talk about is joint work with Shawan Chakrabarti, also at NCBS, and done by my student Suranil Ghosh. Okay, so to begin with, I thought it's best to start with what's usually called the central dogma of molecular biology, which is that DNA goes to RNA goes to protein. And of course, DNA goes to itself, which means DNA can replicate itself, which is the mechanism of inheritance. And genes are expressed differently in different cells, which means that the concentration of RNA and protein can be different in different cells. In particular, you can take genetically identical cells and they can express genes differently so that, for example, if you have bacterial cells or cancer cells and you administer a drug, some of them may survive, and this may have something to do with their gene expression, the concentration of RNA, for example, or the concentration of proteins more usually. What this means is that as cells are dividing, obviously the DNA replicates itself, and that's the main mechanism of inheritance. But alongside that, it is also possible that there's inheritance because the expression of RNA and expression of protein is inherited. So in particular, if you think about gene expression, again, think of this as a concentration of some molecule, and it gets perturbed because of some external factors or because of stochastic fluctuations. Then there are relaxation times associated with each gene, presumably, to return to baseline levels of activity, baseline levels of concentration. And in particular, if you imagine that as the cells are doubling, they perfectly inherit the concentration, so that basically molecules are divided roughly in half. Then these relaxation times, if they're longer than the division times, then that is a mechanism to inherit information that's not through the DNA, but that's through RNA and proteins. And that, of course, is one explanation for why you actually see correlations between sister cells, cells that have a common, that have divided from the same cell, in terms of their ability to survive drug treatments. Okay, what we're interested in is thinking about how to model these kinds of correlations in the RNA active, in the RNA concentration, for example. And of course, these things are very hard to measure, how exactly these things relax back. But you can imagine that there's some distribution of relaxation times for how gene expression concentrations. So you can imagine having their half-life measured in the relevant time scale, which is the cell division time. And those genes which have relaxation times that are long compared to division times basically constitute a form of memory in the cell, which is not genetic memory, but which is non-genetic memory. Okay, now if you imagine that you have a cell that's dividing, goes through a division process, and you look at the expression of each gene in these 4 cells that come from a single common ancestor, if you only have genes present whose relaxation times are non-memory like, which are very fast compared to division times, then the cell-cell covariance matrix, you can imagine, will not have any particular correlations. There'll just be identity along the diagonal, and they'll be sort of random across the other elements. But on the other hand, if you have genes that relax slow compared to the division time, then you can imagine that there's structure in the cell covariance matrix, and that structure is actually informative about the relaxation time of particular genes. And so if you're interested in identifying the dynamics of gene expressions from a single snapshot, like a gene cross cell matrix, then the correlations, the cell covariance matrix actually has information about dynamics, even though it's actually taken at a single time point. Okay, so the kind of data that's available in biology is that at a single time point, you can measure the gene expression. Basically, we can measure RNA levels of every gene in a cell, in several cells, but at a single time point. You have to fix the cells to measure them. You can't measure dynamics, but you can take a single snapshot. And so you imagine that if these cells have some common ancestor, they're related by lineages, there'll be structure in the cell covariance matrix. In particular, right, there'll be sister cells, cousin cells, and so on. And that structure is sort of represented here. So the true covariance matrix in the ensemble will have some particular structure. But typically what you have access to is what's the sample covariance matrix, right? You take a particular sample and you don't know the lineage relationships. They're all mixed up. And so you get something that looks very noisy. So the question we were asking is, can you identify the time scale of the dynamics of gene expression from a single snapshot? Okay, and so to do that, we basically used ideas from random matrix theory. And so the null model for what the cell covariance matrix looks like is the Marchenko-Pasteur distribution, right? And that's given here. It has compact support, as usually you get in random matrix theory. And the parameters are basically only a function of, for an m cross n rectangular matrix, the covariance matrix will have a distribution of eigenvalues, which is only a function of m by n. In our case, the number of genes by cells. So here's what the same distribution of eigenvalues looks like on a log-log plot, where this is log of the rank and a log of the eigenvalues. And so the eigenvalues are, if you have just a random matrix with no correlations between cells, should follow an MP distribution. But now we can try and derive the eigenvalues of a cell covariance matrix, which are actually related by this kind of a lineage process. The true covariance matrix, I'm not going to give you all the details, but the true covariance matrix, if you assume that there's some sort of exponential relaxation dynamics, is sort of easy to write down because of the structure of division. You can also calculate the eigenvalues of the true covariance matrix, and I'm not showing you, but this is not very hard. And then finally, to get the distribution of eigenvalues of a sample covariance matrix, you basically use the Stieltjes transform, in fact, there's something that Marchenko-Pasteur even had in their original paper, that if you know the true covariance distribution of the eigenvalues, then you can find out the sample covariance distribution of the eigenvalues. Okay, here's just to show that you can do this. So you can take, for example, an OU process, and you can construct these kinds of matrices that come from a division process, where you take a snapshot at the very end, and you have things that are fluctuating in every cell. And what we find is that the true covariance matrix basically has degenerate eigenvalues, and which are sort of power law distributed. And if you have slow division, which means again your division time scale becomes much longer than the time scale of fluctuation of all of the genes, then you go back to the MP distribution, Marchenko-Pasteur distribution. Similarly, for the sample covariance matrix, we can actually analytically calculate what the eigenvalue should be, we can, here's a comparison of the numerics with our analytical result. Okay, so that actually suggests a simple algorithm for how we can identify genes that have slow relaxation timescales, which is that if you have a gene cross cell matrix, again, the kind of data that you have access to in biology, then the idea is that if you remove a fast fluctuating gene, a gene that fluctuates at fast time scales, then that gene is going to have a small effect on the distribution of eigenvalues. Whereas if you remove a slowly fluctuating gene, again slow compared to the division time, then that's going to have a large effect on the distribution of eigenvalues. And in fact, the distribution of eigenvalues is going to go more towards looking like an MP distribution. Now this may be a small effect because you're talking, of course, of thousands of genes and removing a single gene, but we are analytically again able to calculate what this effect will be, and it turns out to be significant enough to be able to use it in practical context, given the kinds of numbers involved. So we then, of course, applied this to single-cell RNA sequencing data sets. And first of all, we found that the distribution of eigenvalues of the cell covariance matrix in these data sets are very distinctly non-Marchenko-Pasteur, reflecting the fact that actually there is a lot of cell covariance in these data sets. And it does, at least for a range, look sort of power law-like, although I'm not going to insist that it's a power law. And then finally, using the kind of algorithm that I mentioned, we're also able to identify previously suggested slowly fluctuating genes, which were discovered by much more tedious methods, which I won't go into right now, just again from this kind of a single snapshot. So with that, I'll conclude just to say that I think random matrix theory is actually very usefully applied to the kind of high-dimensional data sets that are now available in biology.
Thank you, sir. Let's have some questions from the audience.
Yeah. Which one? Yeah.
Yeah, here.
This.
Right.
Yeah.
Where do you get the values for the B and the muse?
Yeah. So yeah, the muse are the relaxation times, which you have to assume a distribution for. and the B is the number of branching events, which we don't know. So actually, we are not able to actually figure out the relaxation time of genes from our method, but we are able to give it an ordering. So relative information is preserved, though we don't know exact time scale information, because that depends on the distribution of relaxation time and the number of branchings.
So are you essentially diagonalizing this random matrix or what?
No, so this is the true covariance matrix. What we are interested in the sample covariance. Here you can find the eigenvalues. So yeah, these are the eigenvalues. You can diagonalize it exactly. But the sample is what you have. You don't ever have access to the true covariance matrix because you always only have a single sample. Or I mean, in realistic situations, you have a single sample. And that also you can calculate the eigenvalues for using this kind of transform. Yeah.
And is there any mistake? The 2b mu b is missing in this the second term.
Yeah. Yeah. What is missing?
0 1 element.
Where?
First row, second element.
First row, second. No. So this has a structure where so the idea is that, right, you imagine that you start from a single cell, right, like here, and then you have two division processes. Then these two cells are going to be more correlated than this cell and this cell, because they have a common ancestor which is nearer in time than this cell and this cell. So actually, you need to figure out the number of sisters, the number of cousins, number of second cousins, and so on, and that will change the correlation. So it has a nice sort of structure.
So in the first level, it's 1.
Yeah, exactly.
Yeah, okay. Thank you.
Yeah.
Hi, yeah. The MP distribution is what you get if there are no correlations.
That's right, yeah.
Right? So could you go a couple of slides afterwards?
Yeah.
Yeah. So here, the purple one is for slow division.
Yes.
Slow division implies memory.
So slow division time scale. So what I mean is that large division
Long
time
time
scales,
scales,
which means the genes fluctuate at time scales that are faster than the time scales of division.
Right.
And so you don't have memory of so if you're at a high value, you don't have memory of that by the time a division happens.
I see. Okay, got it. Thank you.
Yeah, so it gives you back very nicely. It gives you back the MP distribution. Yeah.
Where you remove the rows.
Oh yes, yes.
Yeah, here what was the plan?
Say that again.
I mean, because similar things are done for mutations.
Yes. So well, it's not quite a mutation because we're not actually it's just the same data set.
Yeah.
So we don't actually know the biological effects of removing this, but it's more just a mathematical trick to see how much every column contributes to the eigenvalues. And of course, in the limit that this matrix becomes very large, this is going to go down and you can calculate how it goes down.
Okay. So
But,
there's one thing.
Yeah.
That there are certain keystone.
Right.
You don't have to have a full row. You can just have one single element which you remove.
Yes, which can have an important effect.
Which can have a very important effect.
Right,
As a matter of fact, it trashes whatever the thing is supposed to do.
Right. Because there's inherent heterogeneity in the columns that comes from the distribution of time scales.
Right.
Yeah.
So have you checked on that?
Well,
and
so I'm
what
not sure.
meaning would it have in your context?
So again, so maybe we should take this offline because I think so it requires a discussion.
Right.
Yeah.
Due to the time constraints, let's thank the speaker.